I would now like to welcome to the floor our next speaker, Dr. Afshin Beheshti, a scientist at KBR at NASA Amy's Research Center, who will deliver a talk entitled Mitochondrial Dysfunction as being a central driver for health risks associated with spaceflight. Afshin, it's a pleasure to have you back. The floor is yours.
So thanks for the introduction. And again, thanks for having me back. So this is going to be a little shift from the previous speakers, but it's going to have aging in there, of course, but it's going to talk about spaceflight, as Franco mentioned. So before I go on, I know probably most of you audience and then most of you here, the participants know about mitochondrial aging, but just a brief overview. We know that, mitochondria is heavily involved with the hallmarks of aging. I'm sure a lot of you are familiar with this paper that came out in Cell, when was this, back in 2013, called The Hallmarks of Aging. And it's a really nice paper talking about what occurs with aging and all the stuff that the previous speaker and other speakers have been talking about that occurs during aging molecularly. They really outline the steps there and what this review goes over that. And of course, one of the key hallmarks is mitochondrial dysfunction. And as if you're not familiar with mitochondria is the powerhouse of the cells, basically your bioenergetics that keeps you going and keeps you active, keeps you healthy. And of course, if you have mitochondrial dysfunction occurring, this is a older review by Doug Wallace, who's one of the founders of mitochondrial genetics. He in this paper and on many papers after this, we're discussing, basically, if you look at your cells with age, the mitochondrial function goes down. And then this is related with like reactive oxygen species, oxidative stress, and eventually cell death, because, if your bioenergetics and your cells stop working, of course, everything else will shut down. So mitochondria is very important. This is like the overview, simplification of what mitochondria does, and, of course, and related with aging. But I'm sure people here know all about the real biological details of it, which, will go in the context of spaceflight. And of course, every other disease, when you have mitochondrial dysfunction happening, it affects what you're doing. And with age, of course, with the decline you have with mitochondria, this is going to compound factors and you'll have certain diseases related with it. So most people here, I'm not sure if you're familiar with spaceflight experiments and what data is used for that. So briefly, going over spaceflight and the space environment. And so the space environments involved with, there's many different things out there. So in Earth, we're, of course, protected. We got the ozone layer. And then outside the Earth, we have this magnetic field called the magnetosphere, or the Van Allen belt. And right now, all the experiments that are done in space, which is the low Earth orbit, as I said, protected by the Van Allen belt, is usually in that magnetosphere. So the International Space Station, the older shuttle missions that people have done, not just NASA, but around the world, all the other space agencies from JAXA to ESA and other ones around the world, Chinese. And so basically what happens in the Van Allen belt, you got a lower dose of radiation. So in space, you got several hazards. You got the space radiation, heavy ions, these heavy giant particles like iron particles, oxygen, silicon, from things that can happen from like supernovas and Earth are traveling through deep space. And you're exposed to that. And our magnetic field and our ozone layer on Earth, we're not exposed to that. We're protected by it on Earth. And then you got the lack of gravity, as you don't. So the lack of gravity, of course, will impact many different biological functions in your body also. And then you got other factors here, as we highlight here. There's a review we had put out last year in Cell, talking about like the fundamentals of, or what we like to call the hallmarks, of space biology, where you got different things happening like the distance, confinement, gravity, and radiation. But gravity and radiation is what I'm going to concentrate here on mainly, which impacts your health. So just give you an idea, like if you say go to a mission to Mars, which is like the future in a decade or so, a couple of decades, that's the plan, right? To go see what happens in Mars, colonate it, or have people actually fly there and see, explore it in person. So typical radiations you might get on Earth is like, chest X-ray and things like that are very low. And you got to keep in mind the type of radiation we get on Earth is very different than what we have in deep space with the heavy ions. On Earth, we have gamma. So gamma radiation is just like these fast sand particles going through your cells. And as opposed to deep space, is you got these bowling balls sitting and depositing massive amounts of energies in your cells. So the deep space mission to Mars is, relatively speaking, this is this much more radiation you might get compared to everything else, where there's the shuttle missions, or this is an older slide, so Skylab is now ISS, so the ISS missions. So that's a concern, right? A lot of radiation. How does that impact things? And then so the health risks that are involved is basically, females and males, there's a lot of similarities of different health risks you might get. But there are distinct health risks that are associated with female and male, we know. So things like cancer, sleep disorders, muscle degeneration, kidney-related issues, And a lot of things are correlated with aging also. So space, that's another concern in space. Does the space environment, meaning the radiation and the microgravity that's in space, would that, what optimal age group should be sent to space? Currently right now they're usually typical age of astronauts are between 30s to 50s. But should we send older people? What happens if now that things are becoming more commercialized, as we hear about SpaceX and Blue Origin, Virgin Atlantic sending normal civilians up to space that are not at the top 1% healthy population like the astronauts are, or the top 0.001%? Is that a concern? So we just heard recently William Shatner went to space, right? He's 90 years old. So with that age, would it be more beneficial, or than, let's say, sending a younger person, or would it not be, based on maybe acceleration of age due to the space environment? So the types of experiments that we do, just in case you're not familiar with, this is like we send up mice, for example, to the International Space Station. This is a video of mice that are on the International Space Station, our little mighty mousternauts, floating around in their cages. There's a paper we put out just describing the types of experiments that are done. And then there's, the ground experiments, the simulated space experiments we do on ground. For example, we go to a lab called Brookhaven National Lab, which is in Long Island, New York, which is a high-energy physics collider. So NASA had set up a space radiation laboratory there in early 2000s, where you could take those heavy ions produced by these high-energy physics colliders, and then take it like for example, an iron particle or a mixed beam of radiation. That'd be what you might get exposed to, let's say, an hour in space, like iron particles, oxygen, and so on. And then put your samples in front of that beam and irradiate them. So that's some things that I've done and other folks have done who are principal investigators of grants, and they go here and run experiments. For microgravity simulation, like in animals and mice mainly, you have this setup, this is from my experiments, where you hang the mice by the tail, so it's kind of unloading the weight from the back of the leg. So it's kind of like the microgravity event. It's sort of silly, but it's the closest thing we can do to having a simulated microgravity experiment in mice. Another collaborator of mine at Beth Israel, Stuart Rucker, and Mary McTrue in the Boston area, they have these experiments called partial weight bearing rat model, where they have a harness and they could hang the rats at certain elevations to simulate like certain parts of microgravity, like 20%, meaning like the moon gravity or the Mars gravity and so on. So those are the type of experiments. And then let's say if you don't have grants, you're not running experiments, there's actually a treasure trove of data on NASA's GeneLab platform, which is here. This is the site where you can go, and the data is publicly available, where it's all the bioinformatics, all the omics data, basically, genes, transcriptomics, genomics, any type of omics data that's really space is being collected and deposited here. And then GeneLab has made it where the hardcore computer scientists can get the raw data and play with it and come up with novel hypotheses and analysis. And then we also have provided in GeneLab like the higher order data, like the process data. So if you're not, don't want to spend time with starting the raw data, you can start with the process data. And plus, there's a whole visualization platform where people who are not as computationally inclined, they can actually go in the visualizer and type their favorite genes, let's say related to certain aging factors, and see how that might change in these animal models or cell models that are sent to space, or these simulated experiments with omics, and maybe have some novel hypothesis of age that like a previous speaker was showing, that could occur, that maybe space either accelerates or decelerates it. So now the actual story. So we had put out a paper last year that came out in Cell, where we found that mitochondrial stress was a central hub related to spaceflight that impacts the body. And the general overview of this is that, before I go on, I just want to, this is a work done by many different people, and in GeneLab we have these things called analysis working groups, which I run, that's open to the public. Anyone from the public can join, and I lead a multi-omics analysis working group where anyone from around the world can join. We have many people there, and we work on together as a collaborative open science kind of initiative projects that could make an impact. So this project here was basically, I said, when people were involved with this, is there kind of a central switch or central driver in space that can impact your biology? Because in space you have a systemic effect. It's basically your whole body being exposed to radiation and microgravity. So the key is then, looking at GeneLab, we utilize like already available omics data from mice, from like protein level, DNA level to RNA level, metabolites. And then, using our various skills in our group with pathway analysis, modeling, computational analysis, we started like picking out the story that came out. And the story came out over and over again, looking at many different tissues from mice that were there, and also cell culture experiments that were done previously, that mitochondrial pathways were always showing up were key. No matter, and we looked at certain different ages of mice too, no matter the age. And then we also had astronaut data. We had like the largest amount of astronaut data in this paper, physiological data from key targets related to mitochondria that was related, to look at aspects of, in the human model, if it's related to that. And then also, you might have heard, I'll go briefly over this, the twin study data with Scott and Mark Kelly were in space for a year. And Scott Kelly was identical twin to Mark, where Scott Kelly was in space for a year. Mark, his identical twin, was on Earth. So we have data from that, actually, from the blood and RNA sequencing data to confirm what's happening. Granted, it's an N of 1, but it's, kind of a proof of concept thing. So again, how does this relate with aging, since this is an aging meeting, not a space biology meeting, is that, again, mitochondrial being a key hallmark of aging. And what we put out as a review, mitochondrial based on this paper, again, is key hallmark in space biology. There's other factors that are related with space too, which I won't go into this talk, but there's others folks that are, in the field that I know could give a talk about this. For example, telomere length. With aging within the astronauts that are in space, Susan Bailey, who's in Colorado State University of Colorado Boulder. I forget, I always mix those two up, but one of those two. But she's a very prominent PI in NASA World the Twin Study, and what she found is that, as we know, telomeres shorten length with age, right? So, but she found in astronauts when they're in space, starting with the twin study data, and now she has a lot of great data looking at further, the telomeres lengthened. So are people getting younger, or is there something else happening? But when they return, the telomeres get back to regular length. So with aging, there's some things happening that not only in the mitochondrial level, but other factors that we say are like epigenetic changes and so on. So specifically for the mitochondrial story, so we start out that we started looking at unbiased way, tell us what the data, omics data tells from all this data that's available. And again, this is the cell data that we looked at, and the central overlapping pathways that from all, long story short from this, to look at what was happening was all related mitochondrial activity and stress happening. And the majority of these mitochondrial pathways, which are in the green highlights, are downregulated. So it's heavily suppressed in the cells. Just the cell culture in space, coming back and then analyzing it, that was mitochondrial suppression key, in cells. And then once you start looking at the mouse data, these are from two different mice strains, as you see here, Biopsy and C57. You got to just know one little distinction is that C57 mice tend to be more radio resistant, meaning resistant to radiation. Biopsy mice tend to be more radio sensitive. So, that's a key difference. But even though that key difference is there are some mitochondrial expression differences that occur with them. But the overall story is that mitochondrial changes are pretty much universal. Depending on the organ, some organs might not be as prominent for one strain of mice for another, based on what you're seeing here. But long story short, here, like this, there's a lot of data here we did from RNA. This is a kind of a network level representation of mitochondrial activity in all these different organs: liver, kidney, eye, adrenal gland in these mice. And the inner ring is the protein data, proteomics. Outer rings is the transcriptomic data. And again, mitochondrial activity is really being dysregulated. This is the muscles. And the muscle it's going to be interesting how it has the key relationship to the liver. In general, the muscles are being downregulated in a lot of activity you see. And then this is just looking at mitochondrial genes overall, all the mitochondrial genes out there that are available from a source called Mitocarta, which is from Broad Institute, that you'd see that mapped onto the different organs. And you can see like certain organs, like here, the liver is having a prominent role. The liver here is having a prominent role in a lot of mitochondrial dysfunctions happening, which is, makes sense because that's also related to, your metabolism and mitochondrial activity there. And it's all the organs have it. The muscle, interestingly, is the soleus muscle, which is here. And soleus muscle is one of the first muscles that really start degenerating in microgravity conditions. Going further, looking at specific mitochondrial factors, you can see like this is looking at metabolites happening during metabolomics in the muscle. You see all these key pathways, top pathways related to mitochondrial activity being dysregulated. Looking at specific oxidative phosphorylation genes, and then looking at integrated stress response genes, you see, depending on the organ, certain mitochondrial activity happening in a lot of things. And again, don't want to go too much detail, it's a limited time, but I'm going to show you the basic story. This is actually data from now the twin study data. So Scott Kelly went to space, Mark Kelly was on Earth, the identical twins. And what we see here is the ground study, the ground twin, Mark Kelly's here, and the Scott's going to space over time. This is when they're during flight. This is when they come back. When it comes back from flight. And you can see during flight, these are mitochondrial DNA, or genes related to mitochondrial DNA, are being heavily dysregulated in space. Interestingly enough, that when you start looking at physiological data, this is compiled from 60 astronauts from the last few decades, related to mitochondrial activity, like antioxidant factors in the blood, or these factors related in the urine. When these factors in the urine are up, they indicate mitochondrial stress. And you can see this is before flight, this is returning after flight, 30 days after flight, this is during flight, 15 days during, 30, 80, 180 days, so on. And same goes the same for all the x-axis. So you can see there's actually, antioxidant suppression happening, and then these factors here, like the PGF alpha, there it is, increased levels of OHDAG and prostaglandin F2 alpha as biomarkers for cellular oxidative stress happening, which is related to mitochondria. What's also interesting here is now we've done some metabolic flux analysis within the group, where you take the transcriptomic data and then you look at what the metabolic fluxes happen. Now this is starting to be the interesting interplay between the liver and the muscles. So the livers are in the mice are these green and blue. So green is mice that were up in space, and these are the mice that are on the ground. This is now the muscles, where there's the soleus muscle, or there's another muscle here is the EDL. Again, this would be the space flight is here, and then the ground flight's here, space flight and ground. And what we see here is that, interestingly enough, the ground is purple and the space flight's here. So what we see, interestingly enough, is this interplay where that, the liver is trying to be upregulated for mitochondrial function, while the muscle is degenerating and it has mitochondrial suppression. And in the literature, especially with aging literature, this interplay is seen, where muscle cachexia happens with aging. And indeed, when people have studied mitochondrial functions in the liver, they try to overcompensate for this lack of dysfunction, try to get your metabolism up to hopefully overcompensate for this degeneration that's happening in your muscles. But in space, it doesn't seem to be enough to actually help degenerate. And then this is just interesting twin study data, again, looking at the RNA transcript with mitochondrial genes with different cell types. So these are different cell types that we're comparing in Scott Kelly versus Mark Kelly. And you can see that this distribution is going from pre-flight to ground versus coming back, and within time, you can see the mitochondrial genes are redistributing within these cells, which is pretty interesting. So now there's all these downstream impacts. So the functions of mitochondria, and there's a whole bunch of downstream impact on energy and immunity and so on, calcium balance. And one of the downstream impacts is the immune dysfunction. And what we see here is that clearly, the immune dysregulation is being, a lot of cases might be, let me stay on this slide, sorry, is being suppressed. And what's interesting, when you look at the astronaut physiological data, for the sake of time, I'll just quickly skip to here, because the transcriptomic data shows that, you see that factors related to vitamin D or inflammatory factors are heavily, are being overexpressed in the astronauts, while things that are related to vitamin D, which you want to have up, and other things are, your VEGF, meaning your angiogenesis, they're either up or down and being dysregulated. Again, so you go now, mitochondrial and, fatty acids and, your lipid, like homeostasis, is heavily correlated with each other. And this is just a review paper that discusses the nice one, talking about mitochondrial dysfunction and things that would cause for, lipid dysregulation, things that could cause damage to your liver. For example, you could have non-fatty alcoholic liver disease, NAFLD, which would cause lipid accumulation, which we've shown in other papers that occur, which is downstream of what could happen mitochondrial dysfunction. And with aging, these things happen too. So what we see here, long story short, again, is that that the lipid dysfunction downstream of that, whether there's cholesterol levels in the astronauts, you see it coming up, which is a bad thing, or other fatty acid related things are happening within the transcript level. And what's interesting here now is, again, the metabolic flux analysis comparing the liver to the muscle. Same story as before, where the in the muscle, there's heavy suppression with all these things related with either metabolism or glycolysis, things like lipid-related factors are being heavily suppressed, while in the liver, they're being overexpressed, trying to overcompensate what's happening in the muscle. And then there's other factors related like circadian rhythm, with sleep again, with age, circadian rhythm issues happen. Space flight is a really common parallel to that, cell cycle and so on like that, and your cells are behaving and being greatly affected. And one interesting thing is your olfactory pathways are being heavily dysregulated in these mice, and astronauts have reported things talking about, their smell and taste kind of going a little haywire when they're on space and they come back and it recovers. So, there's different factors, and a lot of these things are actually downstream of mitochondrial dysfunction, or all of them are. So in reality, the summary of the work is that what's happening in space based on all this work is that we said, yeah, basically the key thing is mitochondrial dysfunction. And mitochondrial dysfunction is really upstream from a lot of these factors. And the parallel to aging is really interesting because, a lot of the factors I've shown, especially when you see, go back to, let's say, the Hallmarks of Aging paper, it occurs in space are similar. And now to figure out, let's say, is it beneficial to send someone at a certain age, because we already know with let's say when you're 70, you have a lot less bioenergetics and you have a lot more mitochondrial suppression happening as if you're 50 or 40. So is that going to contribute to further health risks? And just I'd like to make a little plug if you're interested in space biology research in general. We had organized, me and a couple other PIs, Chris Mason and Susan Bailey, had organized this large package of cell papers, of space biology papers that ended up in Cell Press. And we got these covers here that actually Scott Kelly provided. As you can see, the images from the ISS. There's 29 papers here, and you can go here and read all about space biology research. And the telomere work that I just mentioned, from Susan Bailey is also here that you can read about. And of course, the Gene Lab folks that are involved in general. So thanks for your attention, and happy to answer questions later.
Thanks so much, Afshin. A delightfully comprehensive and insightful presentation as always. So thank you so much for that. Viewers who have questions and comments for Afshin, please pop those in the Q&A box at the bottom of the screen, and we will address those during the panel discussion. And also, viewers may be interested to note that both myself and Deep Knowledge Group General Partner Dmitry Kaminskiy were co-authors on a paper with Afshin and over 30 other colleagues a few years ago, I think 2017 now, on the topic of optimizing human health for long duration space flight, colonization, etc. And that paper did briefly touch upon some convergences between detrimental health effects of spaceflight and the biology of aging proper. So for any viewers interested in viewing that, it's available on OncoTarget, Vive la Radio Resistance. So thank you, Afshin. I would